Okay, so we continue with the lecture, right? Now we'll be looking at the control gene expression at the translational level, right? So transcription will have occurred to form the pre-mRNA. The pre-mRNA will have gone through post-transcriptional modifications to form the mature mRNA. The mature mRNA is already transported into the cytoplasm. Okay, so the translational control would control whether this mature mRNA gets translated or not, because it's not like once the mature mRNA reaches the cytoplasm, then straight away it will be translated, right? There's some form of control there to decide whether the mature mRNA actually gets translated and when does it get translated. And also, translational control would also control how many times translation can occur for one piece or one molecule of mature mRNA by determining how long this mature mRNA remains in the cytoplasm before it gets degraded. All right. So in terms of the how long the mature mRNA stays in the cytoplasm, that is described as the half-life of the mRNA. All right. So the longer the half-life, obviously that means the more stable the mRNA is, right? It stays in the cytoplasm longer. So if translation is triggered, then it can be translated multiple times and more times, right? As long as it stays in the cytoplasm, it can be translated, and therefore that one piece or one molecule of mRNA can give rise to more proteins. Right? So this controls how many proteins are synthesized or how many copies of the proteins are synthesized with one molecule of the mature mRNA. All right, so the half-life of the mRNA is lengthened or prolonged by the presence of the 5' cap and the 3' poly A tails, because they function to block the degradation of the mRNA by the 5' exonuclease, which cuts the mRNA at the 5' end, or the 3' exonuclease, which cuts the mRNA from the 3' ends. All right. Okay, so the 5' cap, right, actually prevents the degradation of the mRNA from the 5' end by the 5' exonuclease. Right, so enzymes are specific, right? So they recognize specific substrates. Right, so obviously the 5' exonuclease would recognize mRNA as a substrate, or RNA nucleotides, right, as a substrate. So we call what is the 5' cap, right? The 5' cap is a 7-methyl-guanosine nucleotide. It is a modified nucleotide with the addition of a methyl group, right? So that wouldn't be recognized by the 5' exonuclease, and therefore the 5' exonuclease cannot work by to hydrolyze the mRNA. Okay, so once the cap is removed by some other enzymes, which is obviously a regulated process, then the 5' exonuclease can start to degrade the mRNA from the 5' end. All right. So for the 3' exonuclease, right, it will be, or rather the mRNA, right, will be shielded from degradation with the presence of the 3' poly A tail. Right. So the poly A tail is made up of normal RNA nucleotides, right, with the adenine base. Okay, so so the nucleotide at the 3' poly A tail is not modified, which means that your 3' exonuclease actually can go on to start the hydrolysis or degradation of the mRNA. But then it is going to degrade the poly A tail first before it reaches the actual mRNA sequence that codes for the protein, right? So the 3' poly A tail, the longer it is, means that the longer the mRNA can remain intact in terms of the sequence that codes for protein for translation. So a mRNA with a longer poly A tail would have a higher half-life compared to a piece of mRNA that has a shorter poly A tail. Okay? So that's what I'm trying to say, right, with this animation. Yeah, so once the poly A tail has been degraded, then of course the 3' exonuclease will start to degrade the actual mRNA sequence, and once that's degraded, then obviously that affects translation, right? So with the presence of the 5' cap, as well as the presence and length of the 3' poly A tail, that would determine how long the piece of mRNA would stay intact in the cytoplasm, And if translation has been initiated, then this piece of mRNA, the longer it stays in the cytoplasm, the more times it can be used as a template for translation, then the more proteins would be made from this one molecule of mature mRNA. All right, so that's how the presence of the 5' cap and poly A tail controls translation in terms of how many times translation can occur. Okay, so there are other mechanisms to control the length of the poly A tail as well. Certain hormones or certain enzymes can actually cut the poly A tail, and that would quicken the degradation of the poly A tail by the 3' exonuclease, and therefore the degradation of the mRNA. So that lowers or decrease the half-life of the mRNA, right? And some other proteins also can bind to the 3' UTR, right? So the part of the mRNA that's like a downstream of the stop codon, right? And that marks the mRNA for rapid degradation as well. Right? So these are other mechanisms that you may want to know, right? In case they come out in some of the examples or case studies in the exams. All right. Then besides controlling how many times translation can occur and therefore controlling how much proteins are produced, right, we can also control whether proteins are produced at all from the mRNA, and that is to control whether translation begins or translation is initiated or not. All right, so we call your protein synthesis process translation process, right? Your mRNA is supposed to bind to the small ribosomal subunit via the 5' UTR of the mRNA, right? So certain proteins called translational repressors can actually bind to the 5' UTR to block binding to the small ribosomal subunit, and therefore blocking initiation of translation. So I have an example here of a repressor protein that is binding to the 5' UTR. What happened is in this case study or in this scenario, the 5' UTR is folded, and that folding is locked by the binding of the repressor. So therefore, the 5' UTR would not be able to bind to the mRNA binding site on the small ribosomal subunit. No initiation of translation. But besides proteins, repressor proteins, some RNA molecules, which is complementary to the 5' UTR of the mRNA, can also bind to the 5' UTR and therefore block binding to the mRNA binding site of the small ribosomal subunit. Right, so this control whether the mature mRNA in the cytoplasm actually gets to be translated or not. Okay, so that would be the two mechanisms of translational control.